Welcome to Labiotech Tour. This documentary will take place in the San Francisco Bay Area and its well-known Silicon Valley. I'm Pierre-Louis and this is Jean-Christol. In this documentary we are going to outline the biotech ecosystem around here.
The Bay is famous for its natural beauty and for being the best place in the world for innovation, thanks to the concentration in the same place of top-class universities, venture capital and enthusiastic entrepreneurs. Besides the well-known IT industry with flagship companies like Facebook or Google, the Bay is also one of the largest biocluster in the US. It is actually the birthplace of biotech with the foundation of Genentech back in 1976.
We met over 15 companies and institutions, and our goal is to show you how the strong interactions between the different stakeholders make the Bay Area a great place to create and develop a biotech company.
We will start our visit with an institution that contributes to the success of startups in the area, the QB3 Incubator. It is one of the best biotech incubators worldwide, and it is located at UCSF, the University of California, San Francisco.
Welcome to San Francisco and to this wonderful campus of UCSF. This is the Mission Bay campus down here, which we started about 10 years ago. We already have put about $2.5 billion into developing this campus, and it's helped to attract biotech companies and startups to this area of the city that was once very desolate. 10 years ago, we started a very, very novel concept for the University of California, which was to try and take the money that was going into the vast amounts of money going into basic research and fundamental research here, and see if we could convert it into benefits for the citizens of California. And the way we tried to do that was to see, could we actually help startups to form? Why startups? The attraction of startups, you've probably seen the data from the Kaufman Foundation, that almost all new job creation is actually in startups, very little from the senior companies. So if we're trying to create jobs for young Californians, the best way to do it is to do it by creating startups. So it should be a good trip, but make sure you get up to Napa Valley to see if you can compare the California wines to the French wines.
QB3 seems to be a great place for entrepreneurs. So let's now drive to the building where the QB3 startups are located and meet the most promising of these companies.
Welcome to 4D Molecular Therapeutics. I'm David Kern and I'm the CEO and co-founder of the company. We're based in San Francisco, California in the QB3 Incubator Lab space. We're a leader in gene therapy. We create customized and targeted delivery vehicles to cure genetic diseases. And with this technology, we plan to unlock the full potential of gene therapy. The vast majority of hereditary diseases lack effective therapies. So there's a huge unmet medical need for patients with things like cystic fibrosis and hemophilia. Gene therapy has the potential to cure these diseases. But to do so, we really needed three key components. The first was the genetic knowledge of what genes are defective in each disease, and we now have that information. Second, we needed the repair packages that would be able to cure disease cells in the body, and we now have those. The third and missing component is delivery vehicles. So the current products in the clinic are derived from one of 10 available natural serotypes, roughly. And these really aren't customized in any way, so these products are used in a very small percentage of the total diseases that could be addressed with gene therapy. At 4D Molecular Therapeutics, we're solving the delivery problem, and our vectors are selected from a library of AAV vectors we created of over 100 million vectors, so 10 million times more powerful than what was previously available. And with this, we create customized vectors that can address any tissue in the body and really open up the full potential of the field.
Welcome to Biosystems. I'm Brad Rice, Chief Technology Officer. We are in the QB3 incubation facility today in San Francisco. Our technology involves basically a novel method for tracking cells for cell therapy applications. You may be aware of the great promise of cell therapies, stem cell therapy for neurodegenerative disease, heart disease, spinal cord repair, T-cell therapy for treating cancer. But a significant unmet need in all of these areas is the ability for researchers to know what's happening to the cells when they're injected or applied to a human. And so we're trying to work on technologies that will allow us to track and manipulate those cells. We want to do it non-invasively. Right now, technologies require maybe extracting tissue to analyze where the cells are going. So we feel it's important to have a non-invasive, wireless, if you will, technology for tracking cells and being able to manipulate them in vivo.
Hi, my name is Sangeeta Iyer and I'm a scientist at Feldstein Lab. Perlstein Lab is interested in finding cures for rare diseases. Why are we interested in rare diseases? There's about 7,000 different rare diseases that we currently know about. More are being added to this day by day as we have advances in genome sequencing. About 80% of these diseases are inherited, which is to say that they're associated with different genes. At Perlstein Lab, we try to understand the genetic basis of these diseases and then study them using our novel platform involving model organisms.
We are using multiple model organisms, as Sangeeta just mentioned. So we are working with worms, flies, yeast, and mammalian cells, human cells. And we're developing a platform in order to do high throughput drug screening on these model organisms. And we've altered these model organisms to carry patient-specific mutations so we can find precision therapeutics. And we are hoping to parallelize this process such that we can look at multiple rare genetic diseases at once. So right now we are currently working on Niemann-Pick type C, but our next disease in the pipeline is N-glycanase deficiency, which was recently diagnosed, and then we will be moving to other diseases as well once we can scale up and parallelize the process.
Hi, welcome to Hoe Biome. I'm Colleen Cutcliffe. I'm the CEO and co-founder. We have 8 people that are part of the team, and we've been around for about a year and a half. We're building diagnostics and therapeutics which target the microbiome. That's all the bacteria that's on the outside and inside of each person. For a long time, we've known that the bacteria that reside on the outside and the inside of you play an important role in health as well as disease. But only recently have the technological advances been made which allow us to really survey who those microbes are and what functionality that they're playing. In order to really translate this information into diagnostics and therapeutics which can be brought into the clinic, you really need to take a systems biology approach to the problem. And it's this systems biology approach that Hoe Biome is taking.
At the end of this first exciting day, we head back to our Airbnb located in Foster City for a good night's sleep. After a few delicious pancakes, we drive a few miles to our next meeting in Menlo Park. We cannot resist a short break at the Googleplex to try out their bikes. And now we are going to meet a company working on circulating tumor cells.
Welcome to Vortex Biosciences. I'm Michael Boistacino, CEO. Our team of 8 people here in Menlo Park, California are developing microfluidic technology to extract and analyze circulating tumor cells from cancer patients.
I'm Elodie Solier, CTO of Vortex Biosciences. As you may know, cancer is still the first killer worldwide. Why? Because usually the cancer is detected way too late, and also because of the lack of a therapy personalized to each patient, because each cancer is different. So the interest of these circulating tumor cells released in the blood by the tumors is the fact that they give access, they enable the clinicians and the researchers to detect the cancer much earlier, but also to identify the source of the cancer, its aggressiveness as well, and also to predict the effectiveness of a given therapy for each patient. So our instrument is simple enough to be easily deployed in any cancer center or research labs. The user just takes a standard blood tube, connects this tube to a cartridge, and bring the cartridge into the instrument. Then the blood is processed through this microfluidic chip, and the circulating tumor cells are collected downstream, either for genetic analysis, for cancer prognosis, or for even personalized therapy.
We have seen a lot of startups so far, and we are now going to meet a bigger company, Amunix, which has more than 300 employees.
Welcome to Amunix. My name is Volker Schellenberger. I'm one of the co-founders of the company. The company was started almost 10 years ago, and we are in the process of discovering novel biologics which have superior properties compared to existing pharmaceuticals. We developed a very innovative polymer technology which allows us to take existing pharmaceuticals and make better versions of them, versions which act longer in the body so that they can be injected less frequently. We can also make versions of existing drugs which combine multiple activities into one particular pharmaceutical so that you don't have to take multiple different drugs. We keep them together in a fixed ratio. And more recently, actually, we also embarked into using our Extend polymer technology to deliver toxic drugs selectively into tumor tissues. So in that case, we are making pharmaceuticals which contain a toxic payload. They use our Extend polymer and they use other elements like fragments, for instance, from an antibody, and deliver them selectively to tumor tissue. So our goal is to develop pharmaceuticals which can help patients with a number of different ailments. And the most advanced product right now is actually in phase 3 trials. And so our hope is that if you come visit us again in 10 years, you will see a lot of Extend-based products on the market. And with that, I'd like to thank you for visiting us, and I hope you have a great trip in the Bay Area.
Biological aging process is attracting more and more attention as Google created an heavily funded Calico. So we are going to have a thematic afternoon on biology of aging and the future perspectives for innovation. We are first visiting the Buck Institute, one of the leading organizations for research on aging, and then we'll go to Exogen, a startup working on DNA damage, which is closely linked to aging processes.
Welcome to the Buck Institute for Research on Aging. We're a nonprofit medical research institute in Northern California, and our focus is on the aging process. Really what we want to understand is what we call geroscience, which I would define as what is it about aging that contributes to disease. We know that aging is the biggest risk factor for a whole range of chronic diseases that are causing a lot of problems for people as they get older, and we'd like to target aging directly as a means of preventing multiple chronic diseases at the same time. Most of our research is around that concept, understanding the molecular mechanisms that drive aging and understanding why the aging process contributes to disease. I think it's a really exciting time in the aging research field because it seems like helping humans stay healthy longer is something that's palpable, it's something that's achievable, and in a sense it's something that's achievable today. We already know that if you make the proper lifestyle choices, you'll likely stay healthy a lot longer, and you probably live longer as well. Think of it this way, if you cured all cancer in people over 50 just like that, the average life expectancy would only go up 3 years. The reason is that 2/3 of people don't die from cancer. And the third that do, well, you've cured their cancer and now they're going to die from Alzheimer's disease 10 years from now. Our belief is that if we target aging, we get a broad spectrum of protection against multiple diseases at the same time. And I think in mice we're already pretty successful at this. If we can translate that to humans, then I think it's really going to create a medical revolution.
Hello, Sylvain. Hello, how are you? Welcome to Exogen. Exogen was founded about 2 years ago, and what we work on is we're looking at DNA damage in individuals. One of the main reasons why we're interested in DNA damage is because one of the root cause to cancer is the damage that we get all the time on our DNA that doesn't get repaired correctly. And also it's an aging process. And so by measuring this, we believe that we may have the next cholesterol test for cancer in the future.
We have seen a lot of health biotech the previous days. We are now about to visit 2 amazing companies in the bioindustry business. These companies are engineering microorganisms to produce valuable molecules and chemicals.
Hello, my name is Pierre-Louis. I'm the CEO of Hoe Biome Inc. We are located in Sunnyvale, California. Our technology is about conversion of waste carbon into renewable chemicals. Our goal from inception was to try to find processes to produce renewable chemical at cost competitive prices. We try to identify source of carbon that will be inexpensive and very abundant. When we look at the industry, there were already technologies utilizing sugar and cellulosic sugar to make renewable chemical. We thought of finding a different feedstock that will be abundant and inexpensive. So we selected gases and we selected microbe able to use CO2 as a source of carbon and hydrogen as a source of energy for producing chemicals. Once these microbes were adapted to grow on cement flue gas and some other industrial flue gases, and these guys, by the way, are not purified or concentrated. We are using them unadulterated. We modify this organism to be able to produce some chemical or commercial value. We have, for instance, validation and production of N-butanol, bioplastics, and carotenoid from cement flue gas and natural gas combustion from power plant.
Hi, I'm Jamie Batcher, CEO and founder of Pareto Bio Technologies. We're a renewable chemicals company spun out of the Salk Institute in San Diego with technology that's been developed for enzyme engineering over the last decade plus. Pareto is a technology provider for our partners. The technology lets us generate renewable chemicals, and this is really important to our partners and to our customers because they're interested in lower cost, lower price volatility, and green alternatives. That is, reducing reliance on petrochemicals, oil-derived chemicals. Our first application for our technology is going to be in the flavors and fragrances field, where it's also really important that our products are natural. The market there greatly prefers natural products, and that's what we can supply. We don't stop there, though. Just being in flavors and fragrances would be a great business, but going well beyond that lets us have a real impact on the world. And so our next product after flavors will be in food preservatives, and then in a number of different areas, such as nutraceuticals, cosmetics, and other specialty and commodity chemicals.
We want to know more about how these biotech companies are funded. Therefore, we decided to drive to the place where you can find the highest concentration of venture capital in the world. So here we are in Sand Hill Road to meet one of the most prestigious VC firm of the place.
We're now here at InterWest Partners, which is a venture capital firm that's been in business since 1979. This company, InterWest, has broad experience in venture capital. In fact, this is what is called a diversified venture firm, and we invest in both technology, that is software, hardware, and biotechnology and life sciences. InterWest Partners presently is on fund number 10, which means that we've had 10 funds already, and we'll be raising fund 11 shortly. And it has over $2 billion under management. I would say almost 30% of the venture capital money available to US startups or venture firms is right here on Sand Hill Road. And of course, we are here as well. Our expertise here in InterWest is certainly in the area of immunology, and we have been involved with several very successful immuno-oncology companies. So we have put together at least 4 of those companies. Going forward, the strategy we've decided to work on is certainly concentrate in this area because the technology and the science in immunology has become extremely valuable and extremely useful going forward. We've now learned how to begin to think about modulating the immune system. And now new companies which take advantage of all our expertise and all the new techniques that might be available, we are very interested in.
We are now going to the Illumina's office in San Francisco. Illumina is a world leader in gene sequencing, and they also have a startup accelerator. So let's discover one of these startups.
We are Accel Biosciences. I'm Brian Feth. We were founded in 2012, located in Mission Bay, San Francisco, have 8 employees, and are working on a novel diagnostic tool for cancer. To date, biopsies have been really the source of genetic information about a patient and thus how to treat that patient's cancer. However, they're expensive, they're invasive, and unpleasant for the patient. There's an opportunity to really solve this problem using a liquid biopsy, which is really finding information from patient blood. Circulating tumor cells offer all the rich information of the tumor, but from a non-invasive source. However, their rarity has made it very difficult to access these cells in sufficient quantities to understand the full genetic, transcriptome, and proteomic information that they contain. We've developed a novel system that allows us to actually capture and grow these cells into miniature tumors within our system. That really enables us to perform next-generation sequencing on these cells of interest and look at the full genomic, transcriptomic, and protein information from these cells. Enjoy your time in the Bay Area and enjoy your stay.
The Mission Bay area has a great concentration of biotech startups, so we just have to cross the street to go to our next meeting with GigaGen.
Hi, my name is Dave Johnson. I'm founder and CEO of GigaGen Incorporated. We're an 8-person startup in an incubator in San Francisco. We have a technology that captures antibody diversity from deep repertoires, and we express them as DNA, which then we make protein from these libraries to treat diseases such as immune deficiency and treat pathogens such as CMV. We're building a drug which will replace a drug on the market called IVIG. IVIG is a pool of antibodies from thousands of blood donors. And as you'd imagine, this causes all kinds of problems. Risk of viral contamination and contamination from other protein products which are not desired by doctors. Our recombinant or synthetic version will be higher potency, have no risk of viral contaminants, and will not be constrained by blood supply. In the past, when blood products have been replaced by recombinants, this has taken over 80% of the market within a few years, and we expect to do the same.
Our next meeting takes us across the bay to Hayward. We are going there to meet a company in the field of gene therapy.
Welcome to Labiotech Tour. Very happy to have you here today. My name is Jack Lin, CEO and co-founder of Anvil Biosciences. Anvil Biosciences utilizes gene expression drug platform, where we allow the patient's own cells to express therapeutic proteins and biologics. When people think of gene therapy, it's actually quite a wide field right now. Most people think of viral technologies. Those viral technologies are integrative and so are permanent. Our technologies is non-viral, so we think there's a measure of safety built in, and it's non-integrative and yet still can persist for a long time. Our 2 key indications right now are ocular inflammation and neuroinflammation. Right now, they're inadequately treated generally by steroids, and we want to express an anti-inflammatory protein using our technology. This is also a good platform to do it because it is non-viral with a measure of safety, but still persistent and is made by localized cells to affect the area of pathology. Thank you for coming here to the Biotech Tour today, and thank you for hearing about Anvil Biosciences.
For our last stop, we are going to visit the brand new Johnson & Johnson incubator and discover one of the first startups to settle here.
Welcome to Applied Molecular Transport. My name is Tahir Mahmood. I'm the co-founder and chief business officer. We're located in South San Francisco, California at the Johnson & Johnson Innovation JLabs incubator. We are developing products that convert biological therapeutics from injection to pills. Virtually all biological and peptide therapeutics today are given by injection. In addition to the pain and discomfort felt by many patients who have to take these medications, particularly on a chronic basis, there are compliance issues and convenience issues that result out of this, where many of the caregivers, be they family members or healthcare professionals, have to assemble quite often the device packaging and actually administer these shots and puncture the skin to get the drugs into the patient. By converting these medications into pills, we can take away those convenience issues and anticipate that the compliance, the medication compliance to these therapeutics would be improved by the patients. And also we would expect that this would lower the cost of these drugs because we're no longer dealing with the device supply chains that are required, including the cold storage in the case of the injections.
My name is Melinda Richter. I head up JLabs for Johnson & Johnson Innovation. We believe fundamentally that great science is just as likely to come from outside the walls of a big company as it is to come from inside. But the problem is that great science outside the walls of a big company faces many more hurdles to becoming a patient solution or a consumer application. So our goal is to take down those hurdles so the science can succeed or fail on its own. And we do that through two organizations. The first is our JLabs organization. And we do two things there. One, we provide educational and networking opportunities to help build skills and knowledge and contacts between the innovation community within our regions that we exist. And we also provide a physical anchor point, a big company infrastructure for early stage companies so they have the same sort of advantage as our internal R&D teams have. And that ranges from having, within a 30,000 square foot facility like South San Francisco, having half the space being common research space and business space filled with equipment, having an operations team at their disposal, having a business services team that can help them grow. But we also have an innovation center deal team. They work in conjunction with us. They can look at the science and say, what can we do in terms of a creative or a traditional deal construct that can help move that science along? When you believe absolutely that at the core you're there because the patients are waiting for you, then you want to stack the deck for success on the route to get there. And that means going to the place where you have every possible advantage to making that mission happen, where you have the ideas, you have the passion, you have the talent, you have the venture capital, you have the infrastructure, and you have that sense of the patients are waiting. There's no question that's what the San Francisco Bay Area is all about. We couldn't think of being anywhere better.
Thank you for watching the video. We hope you enjoyed it and that you learned a lot about biotechnology in the Bay Area.
And let's keep in touch for our next trip.